So this is kind of a Darwinian example. Kind of looks more like an asexual application of a Darwinian example, where you've got a variant, a mutant variant that winds up in a couple of offspring that wind up being the survivors because the variant allows them to resist the insecticide. So, little iffy. There have been experiments with transfer of, a predator into or out of an environment to change conditions. There was a natural experiment for Darwinian ideas on the Galapagos Islands, ironically enough. In the 80s, there was a couple of the islands had a very long-lasting drought. And the plants on the island are pretty good at going dormant for long periods of drought because it's happened in the past, and then that's sorted out for the ones that could come back. But so on a couple of islands, they wound up with just 2 plants that were still active, were still able to deal with the drought conditions and continue to produce plants. And all the other ones kind of just went out of business for a while. They just went dormant. One plant produced very large, hard casing seeds, and one of them produced seeds that were recessed way down in the bottom of the plant. On this island was a population of finches that ate all the different stuff from all the different plants. But with only these 2 plants available, if they didn't have a big enough beak to crack the seeds, they couldn't use the big seeded plants. And if they didn't have a long enough beak to get down and get the little seeds, they couldn't do that. And what they saw over the course of a couple of years pretty quickly is the population started to radiate, adaptively radiate. And the ones that couldn't do either were dying off and not making babies. The ones that with the big beaks were becoming a population. The ones with the long skinny beaks were becoming a population. Now the drought ended and the dormant plants came back. So it didn't wind up being a speciation event. And pretty quickly diversity returned because it's a sexually reproducing population. And as soon as there was other food that was available, there was no longer selection going on. But it was interesting. It really amazed people how quickly they started to see this settle out in just a couple of generations into these 2 potentially different populations. Something I mentioned in chapter 1 that also came from Darwin is the idea of selection not for survival success, but for reproductive success. What we call sexual selection, even though it applies to asexual reproducers as well. And the word selection here, since it's sexual, people start to think about mate selection, and that's a type of sexual selection parameter. But anything that allows you to reproduce better that isn't directly tied into survival is a sexual selection trait. And sometimes there's a trade-off. Like a peacock display is basically it's a sexual selection trait. The females like the big, showy tails. But it's also a signal of survival fitness. You're basically saying to a potential mate, I have this huge handicap. If I don't get off the ground and fly too well, and I've still managed to survive and make this big tail, and our offspring are gonna have, even if they're female and they don't need the big tail, I've got survival features that they're gonna get as well. So you might wanna mate with me. So there's what they call these hidden signals, even in what appears to be sexual selection traits sometimes. Now we get to accumulated evidence, some of which we've kind of talked about. We talked about homologous features which are built the same, analogous features which are built the same, or which do the same sort of things, and how each of those is related to a particular type of evolution. Is that you've got divergent and convergent. evolution. And that is predicted by evolutionary theory, by Darwin's theories. Is that, sorry, I need to find another pen. So, features selected for under different conditions can split a group over time or even in the same environment and diverge them from a similar ancestor to very different descendants. And it's adaptation to conditions. So is convergent evolution, where the same features are useful in similar conditions. There's certain features that are useful in deserts. There's certain features that are useful in ice sheets. There's certain features that are useful underground, certain features that are useful in jungles. And you'll find a lot of unrelated species having similar features because there's only so many ways to do something. Sometimes you'll see a bunch of different ways, but there's limitations. And so you get this convergent evolution where unrelated species show very similar features because of what the conditions have essentially driven the survivors to. And so you wind up with, these homologous features like the ones we had before. So that's comparative anatomy. One interesting piece of evolutionary evidence comes from the study of embryos and a fellow named Ernst Haeckel, who developed techniques where he could watch embryos develop in living eggs over time. He could open windows up without disturbing the eggs to the point where the whole thing cracked. There's a membrane underneath the shell. He was able to isolate the shell off of the membrane and be able to actually see the embryos. And he watched them develop, and he drew a bunch of pictures, and he sliced them up, and he looked at a bunch of features, and he was surprised to find out that when he compared embryos across a broad swath of, he was looking at vertebrates, backboned animals, but across something like fish and amphibians and reptiles and birds and mammals, early on in embryogenesis, the beginning of embryos, you see a lot of extremely similar features. Convergent evolution, sort of, is that what are the conditions like in an egg? Well, they're pretty similar across a broad swath of eggs. So there isn't a lot of conditional changes that needed to be sorted out. And also, when you change something at an embryological level, it often has a very dramatic change. Eventually, you got to build it. So evolution is kind of resistant to change at the embryological level. I do want to mention something that Haeckel came up with that was, his comparisons were really, really helpful. He had one of these ideas, kind of like Lamarck, that was influential for about 37 seconds, is that people pretty quickly found out it didn't work the way that he thought it did, but I'm going to put it up here because you'll run into it occasionally. I'm going to put it in quotes. At some point in our development, and there's links in the book that show you some of this stuff, we all had open slits here. We had loops of arteries, the way that arteries run through gills. We had a kidney that was built more like a fish kidney. We had a kidney eventually that was more like a reptilian kidney. We did eventually have a mammalian kidney. We had a tail. So Haeckel saw these relationships between the different groups, and he got it in his head that there was a point in an embryo's development where it was not just had gill slits, but it was legitimately a fish. And there was a point where it was legitimately a reptile. And he said, oh wow, this embryo is replaying its evolutionary history step by step by step. It actually is a fish for a while, then a reptile for a while, then it eventually is a mammal or a bird. But it turns out that's not really what you're seeing. You're just seeing similar features in the development of an embryo that turn out to get it where it needs to go. And it never had fully functional gills unless it was going to be a fish. or maybe an amphibian. And, our tails get reabsorbed, because we don't, our group doesn't have tails. But this idea that embryo development replays the evolutionary history kind of got out into the world. Actually, years ago, got an email from somebody who'd read my book online. Not too well, it turns out. He was some rich British guy who was a big contributor to foundations having to do with autism. And he had it in his head that these kids were stalled at the fish stage, and that they could be helped through their development and develop normally if they just chucked them into pools and let them kind of work out, okay, we're not really fish, I guess we need to be mammals. I don't know. It never made any sense to me. And I told him, the whole point here is that nobody believes this. Nobody really believed it for very long at the time, but it was just one of those ideas that held on the fringes and just pops up like he'd seen it. And so I like to tell people, no, that's not the way it works. But comparing embryos across broad swaths of things like not just animals, but plants, watching early developmental stages can show you things are related to one another you wouldn't necessarily expect. Like our closest relatives on our branch of animals, and we'll get to this at the end of the semester, is the group that has the starfish in it. And groups with legs are not as related to us as groups with 5 arms. And you can tell that by looking at how early embryos develop. Part of this has also turned out to involve something called Hox genes. We not only follow similar physical developments, it turns out that there are genes that are responsible for making sure an embryo winds up forming properly. Literally, a gene activates in a spot that goes, okay, this is going to be the front end, this is going to be the back end, this is where the digestive system is going to develop, this is going to be the right side, this is going to be the left side. There are mutations in a Hox gene that actually reverses your side, so your stomach winds up over here and your liver winds up over there and your heart shifts to the right side instead of the left side. But these early formation genes are incredibly similar across a very, very broad band of animals. They discovered a mutant fruit fly. They've done a lot of studies on mutant flies. And there's a mutant fruit fly where instead of developing just their eyes on their heads, they develop little bits of eyes all over the place on their surfaces. And they figured out where the mutation was and went, okay, this must be an eye location gene, an eye location Hox gene. I wonder if there's anything like that in our group. They didn't think so because fly eyes and vertebrate eyes are very different, multiple lenses versus single lenses. And they actually thought that it was a derived characteristic, not an ancestral characteristic, that our shared ancestor wouldn't have had eyes, so it wouldn't have an eye location gene, and it developed separately on the different groups, so they wouldn't be that related to one another. Well, it turns out they are. You compare the homology of the sequence, and you find a very similar gene sequence in mice, and if you disturb it, there are ways you can do that. You don't get mice with eyes all over the place, but you do get mice whose eyes don't form properly. So it is obviously doing a similar job in mammals as it is in flies, and it would indicate that our shared ancestor, that it's an ancestral trait, that eyes are an ancestral trait, lensed eyes at least. If you go into anything that's fairly old, the term Hox gene has changed quite a bit over the last 40 years. It was originally homeo genes and homeo box genes, and then Hox genes, and then capital H, capital O, capital X genes. You still see it that way. And now they've kind of settled on capital H, O, X genes. But this is what happens in biology. It can take a while for people to settle on a particular term. I want to come back to fossils. And talk about formations. So the sediments build up under a body of water, and one way or another, that part gets uplifted, sometimes sideways. And you can now, it's there under the oceans, but you can't really get at it. You want to access these at land once they've come up where you can get at them. And they are full of hard parts, mostly of animals, is that you don't get soft parts or plants much unless the sediments were in a shallow body of water with very low oxygen, where there really wasn't a way to tear apart and consume the dead soft tissue. And there are fossils like that. There's one in the lab. If you look at the fossils in the lab, we have plant fossils in the lab. But there's other ways to encase dead animals to get fossils from them. Sediments are by far the most common. So I'm gonna give you a list, but I'm not gonna write it here. But you got sediments, sometimes sand, sandstone is formed, and things buried in the sand can, compress and mineralize the same way. There actually was a famous fossil discovered. There's a lot of sandstone formations in China. And there was actually a predator dinosaur with his mouth clamped on the back of the neck of a prey dinosaur. Must have been during a sandstorm next to a fairly steep dune, because they kind of went, and the whole thing collapsed on them, and they were preserved in the position that they were. It's still clamped on the back of the neck, and of course, immediately buried, so nothing could tear them apart and get their soft parts gone. And if the sand was originally soft or wet enough, sometimes you get impressions of things like feathers from these Chinese sandstone fossils. And what people are looking for is progressions. The fossil record is very spotty. The sediments are not always continuous. It's a great record for things that live in the water and have hard parts and are common, like clams and snails. Pretty good for sharks that lose teeth a lot, but doesn't really, their skeletons don't preserve very well. But for, like dinosaurs were a land group. So a dead dinosaur needed to wind up in a body of water that became sedimentary rock that we could see it in. And so we don't get the progression that we would like to get of evolution. So this fossil record spotty, and some people go, oh, there's no transitional forms. There's no form that's kind of midway between this and this. Well, that turns out not to be true. There's not, a magical transitional form in every progression, but you get stuff like this, which is actually a transitional form between the dinosaurs, which it was originally classified as before people saw the feathers around it in the imprint. You needed the light to bounce off it just the right way to see the feathers. Between the dinosaurs and the birds. Structured like a dinosaur, but with obviously flight feathers and tail feathers. Archaeopteryx. There's been a decent number of progression fossils found for whale evolution, mostly in India, where the original land animals were very dragony looking, big nasty things. Their closest relatives on land are hippos now, which aren't really I suppose if you recreated one from skeleton, it might look a bit dragony. But we have transitional fossils with much more supportive legs, to the point where the rear legs eventually are gone, as they're totally adapted to the water, and the rear legs, I guess, produce amount of drag, so they gradually went away. Better swimmers, right? Volcanic ash preserves some fossils. Not lava. Lava has two things going against it. It's too doggone hot. It tends to incinerate living things that it buries. And also, it's too hard when it solidifies. So being able to chip stuff out of the lava and get it fossils is almost impossible. But volcanic ash, from a big volcanic explosion, can bury stuff very quickly and leach the water out of it very quickly. So dry it, preserve it. That's how you get fossil footprints, is that footprints in wet sand covered by a layer of volcanic ash, and you get a nasty explosion from a volcano, you can get like this much volcanic ash. Definitely covers and isolates the footprints. It sucks the moisture right out of the wet sand. And so dinosaur footprints, we've actually got footprints of two ancient early human ancestors who are walking along the beach. And you can look at the imprints and figure out that they were walking side by side where one of them had his arm on the other one's shoulder. We assume male and female, but we don't know. Because of the way that the footprints are and how close together they are. So volcanic ash is pretty good for preserving, but it's rare. You don't get a lot of volcanic ash deposits. And of course, amber. There's certain types of trees that defend themselves by producing this very thick encasing sap. And it traps a whole bunch of things, traps bugs and spiders and pollen, and seals them up so that even though the outside of the amber mineralizes, the fossils inside are actually the remnants of the original whatever, the original animal. So if you see a bug in there, actually it's not a petrified imprint of a bug, it's the honest to goodness bug that was there. So those are very interesting fossils of small things that typically would not fossilize also. So they've been really, really good at giving us some idea of what was going on. When you're trying to figure out how old something is, you use the layers. There are radioactive materials that settle in the layers that decay at a rate where you can kind of guess the age of the layers by how much of a certain remnant is there. Carbon dating doesn't go back more than about 50,000 years. So in terms of recent stuff, you can get an age from it. But what carbon, there's not a lot of radioactive carbon in anything that's alive to break down. And so by 50,000 years, it's converted to nitrogen gas. And there isn't enough bit left to measure to know how long it's been continuing to deteriorate. There was a big argument for a while between is evolution gradual or is it punctuated, which means it goes fast and then settles and goes fast and then settles. And at some point, somebody must have gone, well, it's related to conditions. So the rate is going to change. If the conditions change rapidly, then things better evolve or they're going to go extinct. And if rates of condition change is slow, then evolution is going to be slow. So condition changes, or would be if I could spell changes. And how fast do you reproduce? Something that reproduces very quickly can produce a new generation very fast, another generation very fast. So nature can sort through the winners and losers and pass features on to the next generation very, very quickly. When diseases evolve, the things that make us sick tend to have very quick generation turnover, and they, even as asexual reproducers, they tend to evolve faster than we, the hosts, do, even though they do sort out for resistant hosts versus susceptible hosts. There's a video up on YouTube about that for my other class. And basically it comes down to when conditions change and you're looking at populations over time, you got three possible things that are going to happen. The population is going to continue with no significant noticeable change, which means they were okay under the old conditions, they're still okay under the new conditions. Or they're gone, they go extinct. Way more species are extinct than are alive today. Or they change, right? Those are your three choices when conditions change. Either you change in a way that's noticeable, you don't need to change in a way that's noticeable, or you're gone. Okay, that's it for the evolution chapter.